chair that we have there is just starting to hurt my back, so.
Gotcha. All right, well, the plan today is to finish going over the blood, and then to start the heart. I'm still looking, I thought there was a way that I could do a whiteboard through Zoom, but I'm not seeing that option on here, but I'll find something. What I want you to do for lab is the blood typing exercise, the Eldon cards. And so I'm giving you guys a week from today to do that. And so we can go over that. I uploaded a few things about that as well. So why don't I share my screen?
Is this one of them?
Yep.
Okay.
So in our Moodle course, I have the blood typing document, right? And that's exactly what you just held up, Kristen. It's a PDF that talks about these blood typing cards. And then there's a blood typing question. There's one question. It's a Word document. And it's a question about if somebody has this blood type, could they receive blood from this other blood type, that kind of thing. What I want you to do in addition to that is also take a picture of your blood typing card. So I want you to answer this question. And I also want you to take a picture of your blood typing card to see what your blood type is, and just upload all of that. And that will count as your lab for this exercise. So all of that is down here under the blood. Where are we? Here we go. Right, we're going through, finish the blood PowerPoint today, start the heart. So then Friday we'll be working on the heart again, and then probably Wednesday we'll probably finish up the heart and go into blood vessels, and we'll do the dissection sometime next week. All right. Questions?
No, so the case study one, we just answer on that and submit it in, or you want to?
Yep.
Separate paper, okay.
Whichever is easiest for you. It's a Word document, so you should be able to type right on it. It's not like it's a PDF or anything.
Okay.
So you should be able to type right on it, but if not, you can handwrite it, take a picture and submit it that way. You can do however you want. Whatever's the easiest for you is fine with me.
Okay, and that's due next week?
Right, a week from today. All right, so this is basically what you are going to see on your blood typing card. Come on, here we go. So on your blood typing card in your kit, you got a card and it's got 3 ovals on it. One says anti-A, one says anti-B, one says anti-RH. What you are going to do is take the lancet, prick your finger, take a few drops of your blood, put it on A, a few drops put it on B, a few drops put it on anti-RH. And then you're going to use a little bit, I think they just have you use a little bit of water. I think they actually put the antibodies right on there, but I have to look at my kit again just to make sure I know. You're going to use 3 separate toothpicks, one for the anti-A, one for the anti-B, and one for the anti-RH. And you just have to wait a couple of minutes and see if you see clumping, right? And if you see clumping, that means you're positive for that, whether that's A or B or RH. And if you don't see clumping, that means you're negative for that. And what you can do is infer your blood type from that. So for example, if the A clumps, but the B does not clump, you know you're A, right? If the B clumps, but the A does not clump, you know you're B. If both A and B clump, you're AB. And if neither A or B clump, you're O. And then the RH factor, this third one, if you get clumping, you're positive. If you don't get clumping, you're negative. So you can either be A positive, A negative, B positive, B negative, AB positive, AB negative, or O positive or O negative. Okay. So we went through the blood typing, we went through universal donors and recipients, right? And then we were talking about, we still have to talk about the other formed elements that are in the blood. So going back to the beginning of the blood, we said that the liquid part is the plasma, the solids are the formed elements. The formed elements include red blood cells, white blood cells, and platelets. And so we talked a lot about the red blood cells and blood typing. White blood cells are formed elements. There are less of them than red blood cells, but they are larger. If you take any sample of blood from a person and you stain it and look at it under a microscope, you will see only a few white blood cells, mostly red. They help defend the body against invasion. In chapter, let's see, 11, 12, 13, I think it's 13 or 14, we'll talk a lot about how they actually defend your body. They do contain a nucleus and other organelles. So unlike the red blood cells, which kind of kick out their nucleus and their organelles so they can carry oxygen, they do contain the nucleus and other organelles. They don't have hemoglobin. They're not going to bind to oxygen or carbon dioxide. They're only about 5,000 to 10,000 per microliter of blood. And they circulate for a short lifespan in your body. We'll see that some of these white blood cells actually leave your bloodstream and they go into the tissues of the body, right? They become macrophages in the tissues. And some of them we'll see there are these cells called memory cells. So sometimes when you produce these white blood cells, you produce memory cells, which can last for years. So even though most of them only last for a few weeks, memory cells can last for years. If we look at all the white blood cells, we can group them into two big groups, two big buckets, granulocytes and agranulocytes. And it's just based on what they look like when you stain them and look at them under a microscope. Granulocytes have granules in them. Agranulocytes do not. Okay, that's it. That's the only difference between them. That's where the differentiation comes from, is what they look like. Now, there are three types of granulocytes. They are neutrophils, eosinophils, and basophils. There are two types of agranulocytes, lymphocytes and monocytes. Okay. So how do the white blood cells circulate and move? They all can move kind of like an amoeba moves, right? They kind of stretch out and follow the stretched pattern. All of them can migrate outside the bloodstream. So a lot of them can get into your tissues. They can get out of your bloodstream. All are attracted to specific chemical stimuli. One of the things we'll see in the chapter when we talk about immunity is that as your immunity is going on, as some of these white blood cells get activated, one of the things they do is send out these chemical signals. And that attracts other white blood cells to the area to help fight off whatever this infection is. Okay. And some of these white blood cells, particularly neutrophils, eosinophils, and monocytes, are phagocytic. That means they act like Pac-Man. They can chew things up, right? They can chew up things that don't belong. So when we're talking about those 5 types of white blood cells, they are all non-specific except lymphocytes. What that means is all of the granulocytes, neutrophils, eosinophils, and basophils, and monocytes are non-specific. They are going to attack anything that does not look right. Okay, they're not being specific against a particular pathogen, a particular bacteria or virus. They are just looking for things that don't look right. They don't look like they normally should. Lymphocytes, on the other hand, are involved in specific immunity. We will talk in that chapter on immunity. We'll talk about T cells and B cells, which I'm sure you've heard of. T cells and B cells are lymphocytes. Okay, they infer specific to immunity. So they're looking at a particular pathogen. They're looking at the pathogen that causes, chickenpox, right? Varicella zoster. They're looking at whatever one particular pathogen is. That's the pathogen they're being specific to go out and defend your body against. So now, each of the next slides is going to go through each of the different types, the 5 different types, right? Most of your white blood cells are neutrophils. And so over here, the picture on the right, most of what you see are red blood cells. All these little pink things are red blood cells. White blood cells are usually slightly larger than red blood cells. They stain this purplish color. Okay. Neutrophils are granulocytes. You can see little granules in the cytoplasm and in the nucleus. They have a dense nucleus with 2 to 5 lobes. So this dark purple thing is the nucleus, right? You can see 1, 2, 3, 4, 5 lobes. They're usually the first white blood cells to arrive at the injury site because they're the most common, right? They're the most populous. They are very active in phagocytosis, so they chew things up. So they attack and digest bacteria. Again, they're non-specific. They're looking for things that don't belong. So bacteria look different than human cells, so they chew them up. They have a very short lifespan, only about 10 hours, right? It decreases to 30 minutes or less when actively engulfing debris or pathogens. So all that means is they can circulate around your body for about 10 hours before new ones take over. And if they start digesting something, they're only going to last for another half hour. Now, eosinophils are another type of granulocytes. They are similar to neutrophils. There are far less of them in your white blood cells. And their granules, which you can see in the cytoplasm, stain red, right? So here's the neutrophil, here's the eosinophil. So they're red, sometimes they're almost an orange color. That's really the only difference that you'll notice. They have a similar shaped nucleus, they have granules in them. They are phagocytic, but they attack through exocytosis of toxic compounds. So they can chew things up, but mostly what they do is they release these toxic compounds to kill cells. The numbers of eosinophils increase when you have a parasitic infection or an allergic reaction. So parasite, like a tapeworm, you would make more eosinophils that would attack that tapeworm. These also release enzymes that reduce inflammation, right? So they keep inflammation from spreading to nearby tissues. They're anti-inflammatory. And then the third type of granulocyte is basophils. They're a little bit smaller than neutrophils. They're really rare. You don't see basophils very often. They have a lot of granules and they stain a really deep purple or blue, right? So they're the darkest. So the eosinophils are more reddish or orange. Neutrophils are kind of a light purple. These basophils are really dark purple. They're not phagocytic, right? They migrate to the injury sites and they release chemicals. They release heparin, which prevents blood clotting, keeps the blood flowing. They release histamine, which is inflammatory, right? It does dilate your blood vessels. It causes you to have a runny nose, that sort of thing. So these enhance inflammation. And usually the inflammatory response that you get from these is what makes you have a lot of the symptoms that you have, like when you have a cold, right? You get that runny nose, you get the sore throat. A lot of that is due to inflammation. And it's because cells like basophils are releasing histamine, which is why we often take an antihistamine as an over-the-counter remedy. Then we get into agranulocytes. Monocytes are agranulocytes. Right. Now, this is considered an agranulocyte. To me, it still looks like it has speckles in it, right? It's to me, that's hard to tell. Monocytes are really big. If you look at those compared to the red blood cells, they're much bigger than these other white blood cells. So they're really big. They usually have a large nucleus that's like bean-shaped, kidney bean-shaped. They're about 2 to 8% of your circulating white blood cells. So these are kind of, they're somewhat common. They're more common than eosinophils or basophils. These migrate into tissues and become macrophages. So they are very aggressive phagocytes. They look like this when they're in your bloodstream, but they get out of your bloodstream and into different tissues, your skin, your lungs, all these different tissues, your muscles, and they become what are called macrophages. And macro suggests large, phage is eating. So they become these large cells. I think of them like large Pac-Man cells that go up and chew things that don't belong, right? They go around and they chew up things that don't belong. And then finally, the fifth type of white blood cell. These are the specific white blood cells. They're lymphocytes. Now, lymphocytes are usually not very much bigger than a red blood cell, and their nucleus takes up almost the whole cell, right? They have a big round nucleus that takes up almost the whole cell. These are the second most common type of white blood cells. First is neutrophils, second is lymphocytes. Large numbers of them migrate in and out of peripheral tissues. Some can attack foreign cells, others secrete antibodies. T cells and B cells are both lymphocytes. Okay, when you hear about T cells and B cells in your immune system, they are both types of lymphocytes that we'll talk about. So these go out and they actually look for a specific pathogen. They're looking for a particular pathogen that you'll see that they've gotten the message to go out and attack. So sometimes when you go to a doctor and you're, trying to figure out what's causing your sickness, your illness, they want to do what's called the differential white blood cell count. So they not only do they want to, look at your blood and see if you have more white blood cells than you normally would, right? Which indicates that your body's fighting some sort of infection. But they want to specifically see how many of each of the types of white blood cells you have, right? Because certain ones fight allergies more, certain ones fight parasites more. So they can do a test where they can actually take white blood cells and they can estimate the percentages of the different white blood cells, right? You should have neutrophils, then monocytes, then neutrophils, then lymphocytes, then monocytes, then eosinophils and basophils. White blood cells form in the bone marrow, just like red blood cells, right? So you have these stem cells in the bone marrow that when they get certain signals, they become blood cells. They can become white blood cells. They can become red blood cells. Some of the white blood cells we'll see actually go to your thymus and they mature in your thymus. Some mature in your bone. But we'll talk about those in another chapter when we talk about T cells and B cells. Then we get to the third type of cell fragments, or the third type of what do we say? Plasma and what do we call the solids that are in the cells? I forgot now. The solids that are in the blood. So we have red blood cells, we have white blood cells, we have platelets. Okay, those are the other solids that are in the blood. Platelets are involved in blood clotting. Now, in non-mammalian vertebrates, so other animals that aren't mammals, platelets exist as nucleated cells called thrombocytes, but not in us. In us, they're cell fragments. So we have these really big cells, megakaryocytes, that break into fragments. And those fragments are platelets, and the platelets are involved in blood clotting. So if you cut yourself and you don't clot, what happens?
You just continue to bleed.
Continue to bleed. And what happens if you continue to bleed?
Blood loss.
Blood loss, which leads to?
Isn't that like, I want to say anemic, right? Or there's a condition, right? Anemic.
You could be anemic. You could turn hypoxic, right? You're not getting enough oxygen around. Eventually you'll die, right? Because you're not getting enough oxygen. The main thing that I want you to remember is that the blood is carrying oxygen. And so if you don't clot, if you continue to bleed, then you're not carrying oxygen around your body. That's the importance of platelets, right? And platelets only circulate for 9 to 12 days. They don't last for a long time. There's your normal platelet count, right? And if you have a low count, it's called thrombocytopenia. If you have a high count, it's called thrombocytosis. Blood clotting is called hemostasis, not homeostasis, hemostasis. And it happens in 3 phases: the vascular phase, the platelet phase, and the coagulation phase. So if you damage a blood vessel, the first thing that happens is called the vascular phase. What happens is the blood vessels actually contract, right? They get small. So wherever you damage the blood vessel itself has smooth muscle and it shrinks down. And it does that to try to prevent more blood loss, right? So that's the very first thing that happens. It's called vascular spasm. It decreases the blood vessel diameter so that you're not leaking as much blood. So the first phase is the vascular phase. The second phase is platelet phase. It occurs within 15 seconds of the injury. What happens is the platelets, which are in the blood vessel, they actually stick to the cells that are lining the blood vessel. They are sticky, right? And so they start to stick to that lining and they form a little plug. So they kind of plug up that hole, that injury that's there. This phase, the second phase, may even be enough to close if it's just a small break, like a small cut. And the third phase that happens is actual blood clotting or coagulation. Begins at least 30 seconds or more after your damage. There's a whole biochemical sequence, right? There's a whole, it's almost like dominoes, like A activates B, activates C, activates D, like that. That the eventual last step is that you convert fibrinogen to fibrin. Now, fibrin are these fibers, right? These long threads. And what the fibers do is they make this meshwork over the injury. So you've got an injury, right? The vessels shrink down, the platelets kind of gather at that injury site, and then the fibrin makes a meshwork over that injury site so that, excuse me, no cells can leak out of it.
So it's like scabbing, like the scab is the.
Right, that's the scab, exactly. So here you can see, right, vascular phase, if you damage the blood vessel, the vessels shrink, right? They pinch down. The platelet phase, these are the platelets, right? So this is your damaged vessel. The platelets kind of gather up here. So the platelet phase happens with the platelets gathering up. The third phase, coagulation phase, will be kind of a meshwork coming down on those platelets to just secure all those platelets. It's basically just forming the scab so that no more blood leaks out. Okay. The clotting process requires clotting factors. There's 11 different plasma proteins that are clotting factors. You don't need to know them. But what happens is we have these proteins in the body that are converted from proenzymes to active enzymes. Basically, it takes something that's this long and it cleaves part of it and now it becomes active. So fibrinogen is a proenzyme. You cleave off the ogin, right? And it goes from fibrinogen to just fibrin. And that becomes an active enzyme, right? That becomes something that's active. So they interact in a sequence called a cascade. Again, that's like a domino sequence of A activating B, activating C, activating D. Blood clot forms as a result of extrinsic, intrinsic, and common pathways. Now, all right. Extrinsic pathways, that's pretty easy to think about, right? Outside the body. You cut the blood vessels, you injure the blood vessels somehow and blood clotting happens. Intrinsic is more dangerous. Intrinsic means from within. So sometimes high blood pressure, just pushing against the inside of your blood vessels can cause injury, and clotting can happen inside your blood vessels. That's a bad thing, right? You don't want a blood clot inside your blood vessels. That would stop blood from flowing through there, or at least slow it down. And what we'll see is that the extrinsic and intrinsic pathways are slightly different, but there's a commonality to them. So they start off different, right? But they both kind of converge to the same place. The extrinsic pathway is shorter and faster, right? That makes sense. You want this to happen very quickly. And again, I'm not interested in knowing all the different steps of this pathway. The intrinsic pathway starts in the bloodstream. So again, that's when internally you have a reason that this pathway gets started. You're damaging these blood vessels from inside. The common pathway is that whether it starts as intrinsic or extrinsic, it ends up at a common pathway. And that common pathway activates factor 10, okay, which activates prothrombin activator, which converts prothrombin to thrombin. You don't need to know what factor 10 does, right? But what this is going to do is cause clot formation and rapidly prevent blood loss. Now, again, if it's extrinsic, that's a good thing. If it's intrinsic, oftentimes it's not a good thing. That's not what you want to happen. Okay. All three of these pathways require calcium. You need calcium for blood clotting. You need vitamin K to synthesize these clotting factors as well. And here is just a micrograph of what would actually happen. So you can see the red blood cells that get trapped. All of these over here, these little white dots, are platelets. They're pieces of cells. And then all of this, what looks like thread, that's fibrin. So fibrin creates this meshwork, and it holds back all those red blood cells so that you don't lose those red blood cells. Remember, these red blood cells have oxygen bound to them. So the more of them you lose, the less oxygen you're getting to tissues of your body. Clot retraction and removal. So this fibrin network traps your platelets and red blood cells. And then during the repair of the tissue, that clot dissolves. You have other enzymes that can actually start dissolving the fibrin and the other clots. So once once the cells of the blood vessel regenerate, you can dissolve that clot. Questions about that stuff?
So the intrinsic pathway of blood clotting, that's like you said, like blood clots develop like internal, like if they had like internal bleeding type or
So
internal,
not
bleeding,
internal.
but I mean like you said, the blood clots form in like the arteries and stuff.
Right, blood clots forming in the blood vessels is internal,
Blood vessels.
is intrinsic.
Okay.
Internal bleeding would still be extrinsic, right? You're still damaging a blood vessel and it's bleeding. It's just not bleeding outside of your body. It's bleeding into your abdominal cavity or something.
Yeah, I was thinking something like else, but yeah, so like the intrinsic is more like blood clots.
Right, blood clots that form inside the blood vessels that, then becomes a problem, becomes much more of an issue. Because it's going to inhibit blood flow, or it's going to, here we go. It's going to inhibit blood flow, or it's going to completely block off blood flow, depending on where this is. Now, the next chapter is the heart. So we've got the blood, next chapter is the heart, then the blood vessels, then immune system. All related, right? It's 4 chapters, but it's all related. One of the most important things that I find when doing these chapters is that if students understand the flow of blood through the heart, right? If students understand the flow of blood through the heart, they will have a better time understanding exactly how these chapters work. Now, what I'm going to do is actually get off here because it's saying only got 10 minutes left. I'm going to get off here, but we're going to start the heart. We're going to start by tracking the flow of blood through the heart, oxygenated and deoxygenated. So I'm going to get off real quick and then get back on with the same meeting code. I don't know why it makes me do this. And we
were supposed to fix that.
I know they were supposed to fix that and they didn't yet. So we'll figure it out. And then we'll go ahead and we'll draw on the whiteboard. I'll try to draw on the whiteboard and draw a heart and some lungs and blood flow through the heart. All right.
All right.
See you in a minute.